My name is Melanie Samuel. I'm an assistant professor at the Huffington Center on Aging and the Department of Neuroscience at Baylor College of Medicine in Houston, Texas. So in order to describe my work, I thought I would start with a story. This is a story about Wayne and Wade, twins born in the fall of 1929. Now, they were identical, in fact, so identical that their own mother had a hard time telling them apart, and they went on to be inseparable. Now, as they grew up, their lives took a dramatic divergence. In their 50s, Wayne unfortunately suffered a massive brain aneurysm and died. And Wade, on the other hand, went on to work as a carpenter well into his late 80s. So the reason I've told this story is twofold. First, of course, there's a personal connection. Wade is my grandfather. But the second reason is that it illustrates a question. Now, this is a question that has driven some of the best literature that we have produced as humans, some of the best art, and I would argue also some of the best science. And that question is, what makes us unique as individuals? And if we understand the molecular processes that drive this, can we in fact change our stars? Well, as a neuroscientist, I view this question from the perspective of the brain, because it is, of course, our brains that give rise to all of our thoughts and all of our behaviors. Now, brains, in turn, are comprised of building blocks, neurons, that also have their own unique identities. And these unique identities give rise to the emergent properties of the brain that allows all of our thoughts and our behavior. So in our lab, we really seek to solve existential questions for neurons: who they are, how they get here, why they exist, in order to be able to perhaps change the stars, change the fates of these cells. If we can understand the molecular processes that drive neuron identity and neuron function, perhaps we can prevent neural disease or even reverse it. So in order to understand the genes that control neural identity, we really need to study them individually. So the International Mouse Phenotyping Consortium, or IMPC, recognized the need to make animal models in which we could selectively inactivate the function of individual genes in order to study what they do and how they work, so that we could someday apply this information to humans. So the IMPC has generated a large cohort of mice lacking genes that code for specific proteins, one by one. We have used this resource to try to begin to understand and discover new genes that regulate neural identity as well as neural connectivity. It's these connections, or synapses, that allow neurons to communicate with each other and form circuits that in turn underlie behavior. So using this resource, we study a part of the central nervous system called the retina. This is the visual circuit that first detects light and enables vision. We use the circuit because it's well understood and it's tractable, meaning we know how the neurons connect in this system. Therefore, we can ask if these connections are perturbed in particular mutants. So we have used this system then to test individual genes and assess their contribution to neural identity and connectivity, and have discovered several new genes that control neural function. And our ongoing work is aimed at dissecting how these genes impact neural development and also human disease, because it turns out that in several cases, these genes are mutated in rare forms of human neurologic disorders. So we hope that by understanding this process in animals, it will shed light on the pathways in people, so that then we can use this information to perhaps someday develop therapies that prevent disease or reverse it.